Hi, good morning, and welcome to the ZP Developer Zone. So we do this Developer Zone a couple of times a week, and it's really just answering technical questions that have come in during the week. And we kind of try to illustrate some of our answers with obviously images, but also hardware that I might have in the office as well. That said, let me go forward quickly and say today is the 7th of March 2024. And as I say, we try to wrap up questions that have come in during the week, and I'll essentially jump straight into it. It's probably worth saying that a lot of the technologies that we'll talk about today, we will talk about the Sensi-All platform, which I'll just grab. So I will mention the Sensi-All platform a few times. This will become super relevant to soil health and also nutrients as well. Somebody's asked a lot of questions about nutrients, so I will talk about the Sensi-All platform, and we'll also talk about wearables, continuous glucose monitoring as well. So this is a CGM system for continuously monitoring glucose, but I will talk about that. But everything we do is we describe it as kind of sensor to API, sensor to cloud. So I'll try and illustrate what I mean by API in a bit, but application program interface and sensors. At ZP, sensors can start in several form factors. These are sensors, we kind of call them IVDs, in vitro diagnostics. Or this is a sensor, for example, on the back of this watch. This is a microneedle sensor for continuously monitoring. Before you start asking lots of questions about these kind of sensors, to develop work like this is 10 times the cost of developing work like this. So be careful before you sort of start jumping into a big wearables program. When I say wearables, I mean wearable biosensors. Anyway, let me go forward a little bit quicker and say that everything we've tried to do is develop a technology stack that goes from the sensor all the way to the clouds. Our business model is OEM, which means that we basically kind of partner with other companies to put them on the market. But partnering does mean that there has to be some sort, they basically need some form of not income, but money in order to afford all the engineering that might otherwise have to happen. This is the Sensi-All. This is a big part of what ZP does, which is really turning ideas into tangible hardware. And it is tangible because it's not just an image on the camera. It's an image that exists in it's an object that exists in my hand. This is really a fully functioning instrument that can do a lot of things, and I'll talk about that multi-analyte capability in a bit. Our idea is simple enough that people spend too much time developing or thinking about developing technologies for people, and actually with the hardware platform that we have, people can spend more time actually doing and interacting with end users rather than just sort of spend years developing technology that ultimately might fail. And everything we do is cloud connected, so it means that if you're measuring vitamin C in some sort of product, then that vitamin C data can actually go to the cloud. You can see it in real time, and then it means you can kind of iterate and improve, because there's nothing like getting data, even if the data is not the world's best data. Getting something with real customers or real end users is really valuable. Too many people spend too much time not actually reaching that goal. It's probably worth saying on the 26th of March, so not too far from now, in 19 days that is, we are doing a webinar, one in the morning and one in the afternoon. If you are interested in electrochemical sensors, could even be CGM, continuous glucose monitoring, which has come up a lot over the last few days, come to that webinar. So this is on the 26th of March. If you see a link pop up, I'll make sure I put the link underneath the this video as well. It's worth saying that ZP does actually do what we call IVD workshops, in vitro diagnostic workshops, and we also do wearable or CGM workshops as well. These workshops tend to be in Norway, but for bigger clients as well, we do actually come on site and do workshops for them. Now let me get into questions. So question number one, somebody wants to do multi-analyte detection. They want to do sodium and potassium, for example. So I did look it up, and didn't look it up. Sodium and potassium are on our website. I will discuss that shortly. Magnesium is not something on our website, but I believe is doable. Phosphorus, I did look up the definition of phosphorus, and I really should answer this question when it gets to it. But that said, they have a list of analytes. I went through the list of about 70% of the list, and I will discuss that. Somebody else is interested in soil health, NPK, nitrate, phosphate, and potassium. which is ironic because when you look at the first question, somebody's interested in measuring these kind of nutrients. And well, they call it phosphorus, but I looked up and phosphorus means phosphate in this context, and they're also interested in potassium, which was also in the first question as well. But NPK is about nitrate, phosphate, and potassium in the soil. But it's interesting that obviously these kind of analytes or elements appear. They're not elements; they're compounds. Well, some of them are elements and some of them are compounds, but they appear in both questions. So it just shows you how kind of universal, biology is and how life is, because the same nutrients and analytes are appearing everywhere. Quisiera saber el precio de diseño continuo de glucose esta disponible comercialmente. This person is interested in knowing whether our CGM technology is actually commercially available. And it is the quick answer, and I will link to that. They also want, I think I'm mixing up a few questions here, because somebody wants to know whether our technologies will connect to connectors like this. Now, not this particular connector, because this connector is a Dexcom connector. So I have a Dexcom sensor. So of course we don't completely align with them, because Dexcom will have their own intellectual property with the way their sensor connects, and so you've got to tread carefully with this kind of technology. But it's probably just worth saying in passing now that Dexcom, Abbott, and Medtronic are kind of at least in the US and Europe, are the leading CGM manufacturers. And at ZP we do have this kind of wire electrode that you'll see a picture of the Abbott technology in a bit, but we do have that kind of technology. Somebody else was asking about our formulations, and I will touch upon that as well. So question number one, they have lots of analytes, and I did, some of them I had to go away and make sure that, we could, because phosphorus is an element, but I was thinking it's not the phosphorus element they're interested in, it's probably some sort of molecule of it. So I did look up phosphorus, and they mean dipotassium phosphate or disodium phosphate, and then this phosphofactyl dicholine, or this phosphate dicellerine. I should be better. I'm a chemist at pronouncing these, but these must admit, the last two were new to me. But it's worth saying that we do have a phosphate sensor. I then looked up some of the other things, like this riboflavin, which is vitamin B1. I think the niacin was vitamin, maybe it was the thiamine that's vitamin B1, and the riboflavin is B2. So I did look up the structures of these various targets. Its niacin was B3, in fact, and folate is folate. So I did look up the structures and determine that, in fact, just structurally, most of these are electrochemically active. The question rather is not whether you can sense them. The question is, can you sense them in the matrix of interest? So what I mean by that is, all of these molecules are intrinsically electrochemically active. So you can actually, well, vitamin B3 is the worst, B1 is definitely, B2 is, and folate is definitely electrochemically active. So they're electrochemically active, so that means we can detect them quite simply. Question then really is about what's the sample matrix? Is this the human body? Is this vitamin supplements? And then that sort of then drives a question, depending on the application, what's the limit of detection that's required? So sometimes the questions are quite, sort of simple on the surface, but then actually there's, what are the other interferences? What's the specificity that's required? And what is the limit of detection? These become the kind of pertinent questions. Just to say, some of the targets they were looking for were things like sodium and potassium and phosphate. And we do have those, for example, in our standard biosensors. And going into it a bit deeper, they had retinol or vitamin A in there. They also had vitamin B6. So I did go through many of these, getting down to, I think, vitamin B12 and even ascorbic acid. And I'm pretty sure, I did look at vitamin D as well. Vitamin D looked a little bit trickier. So I kind of going through the list thinking, you call it a roadmap. Some of these are probably much easier than others. And easy is, you have to kind of rank them basically based on the idea of what's the easiest to do in the application of interest. And some of these are, fairly easy depending on the application of interest and depending on that limit detection. Some of them look a little bit harder, but we did go through the list. I mean, vitamin E in particular looks fairly straightforward. And I think there was vitamin K was in there as well. Yeah, so I looked at vitamin K as well. So I don't want to oversimplify it. And then I looked up the last one as well, this pantothenate. And I can see that it reacts with something called ATP, adenosine triphosphate. So that gives us a handle on how to potentially detect that molecule. So all that said, you want to do multiple analytes. Some of these analytes are either standard biosensors from CP or fairly simple to develop assays, but really the application is everything, because the application will derive what's the matrix, what's the sample around this, what are the interferences that are probably there, what's the limit of detection, is this essentially a wellness application or a medical application, which will then drive the regulatory requirements. So the quick I must admit I haven't answered the monounsaturated polyunsaturated fats question here, but if I think there are at least cholesterol sensors out there that are electrochemical in nature. If we were doing this, we would probably do it on the Sensi-All platform. And I'll talk about the philosophy of that Sensi-All platform actually now. So essentially what we have is, for every sensor or for every assay, there's a QR code. If you want, just watch a bit more of the slides now, and that QR code will start to become explained as we go along. There's a hardware meter, so the meter will drive those sensors or drive that particular assay. The hardware knows what to do because there's an app. If you're watching this on some sort of screen, it's worth getting your phone out, because you can scan an app and actually put the app on your phone in shortly. And there's a database called Julie. It's a cloud system where we send all of the data. So the sensors work with the hardware. The sample goes onto the sensor. The hardware is driven by the app. The meter or the hardware gives the data back to the app, which actually then stores it on the cloud. But the person also gets a result on their phone. But we actually get all the raw data up in the cloud. This is really important because when you're developing technology, you really want to see what's going on. I'm supporting clients around the world, and it really helps that at any one moment we know exactly where the raw data actually is. It's all stored up in the cloud, and that can be shared with people like myself so that we can help troubleshoot. There is an API associated with Julie, an application program interface. So that means that somebody can sort of dedicate more of their efforts to the cloud and the user experience and the data analytics. If I look over my shoulder, it's just because I'm checking a 3D printer. Yep, I need to just pull something very quickly. Right, back. Yeah, so we can put we have an API because it really allows people to kind of focus on, I think, the bits that are important, because a lot of this hardware for running in vitro diagnostics is actually quite generic, and everyone keeps inventing new hardware which actually wasn't necessarily required or didn't add a lot of value. Here's sensors, here's a meter, sensor goes into meter, it all clicks together quite nicely. That's quite done, and people keep redoing it, they keep reinventing the wheel. Everyone's using Python these days, Pi, Arduino, but they've made it easier to invent this kind of prototyping, but it doesn't actually add an awful lot of value. The value is really the kind of actually getting the product to the market is where you start to add the real value, not the whole development cycle on the way there. We have a bit of a thing called the 2-year startup manifesto at ZP. If we look at the spend that most people spend trying to develop technologies, they spend this money, or they raise money, they spend a lot of time on technology. Because they try to develop the technology, they don't even think about the manufacturing. But obviously, manufacturing meters that you can just send out to people is not so straightforward. And once you've done it, and then you can sort of repeat the efforts. I think it's one thing is worth saying. When you ship out hardware like this, there's little details like there has to be a manual to go with it and stuff like that. So people. They develop technology, there's user experience through the manual, you have to be able to manufacture these things. We are ISO 13485. So they put a lot of effort into technology, a little bit of effort maybe to manufacturing, and probably I think most startups put relatively little effort into business development, which is actually, really the engineering of how I'm going to turn this into a sustainable business that actually makes money. There's not much effort going into that. So they come up with all these technology readiness levels, and they spend, sort of the investors' money or the government's money, doing that essentially. And they may tell investors that it'll take them 5 years. In reality, it'll actually take something like more like 10 to 15 years. And unfortunately, because it takes so long, and people are really probably solving the wrong problems, they're thinking more about technology and maybe then manufacturing. I'm not really thinking whether anyone really wants this technology. The failure rate is 90%, so the success rate is therefore sort of 10%. At ZP, we say, this needs to be reordered slightly, that people need to spend more time thinking about the business. Manufacturing is something that we can already do for them, and technology to a certain degree has already been done. So we want to sort of, put the eyes on the business side of things and slash that 10 to 15 years down to 2 years. It's a kind of MIT, Massachusetts Institute of Technology idea, really, but you have to sort of fail fast or be successful fast. But spending 5, 10, 15 years slowly failing is a little bit unfortunate, because it can be a big chunk of somebody's working life, and then at the end of it, they may not have actually been successful. Well, how do you truncate 5 to 10, 15 years down to 2 years? Well, you have to take care of basic principles. So basic principle, here's a handheld meter. It is already manufactured and shipped around the world. Technology concept, if somebody wants to make a multi-analyte system that takes electrochemical biosensors, there's the concept. Proof of concept means, for me, you've actually done some lab work. That means that you can confirm that you've done vitamin B1, etc. But I'll just go to system prototype. That's definitely beyond the system prototype. System completed and qualified. This system is qualified to do electrochemical sensors and assays, and the system is also deployed. We do ship this, so it's also deployed. It does leave a little bit of science to be done. Proof of concept, it's got to be validated. Because what it is when people talk about multi-analyte detection, the real value add is actually, it is in the business side of things. Can you really make a business from this? Can you really make money from it? But there is also a value add in actually making electrochemical assays. The money or the value is really in the science. It's not in the engineering of doing. You can always find electronic engineers. You can always find software engineers. That kind of engineering is quite deterministic. The effort that you put in is what you get back. Whereas science, you could put effort in and still not be successful. So you are better off trying to add value in these scientific endeavors rather than the engineering endeavors. And then the technology is validated in a relevant environment and demonstrated. We've also taken care of the whole idea that actually there's potentially a customer. You may even get to the point where you're making sales, and then at which point then you find yourself having to have sort of 3 to 5 people just to do logistics. Whereas if you're on the ZP Sense It All platform, in fact all those logistics are taken care of, because you can just place an order with us. We'll put it in a box and ship it to the required people. So that's kind of like an Amazon, dropship model, but it really speeds up the deployment of technologies. The question is, why are we trying to speed up all of this process? Because the reality is there's a horrible surprise waiting for people. That you can sell a technology to enthusiasts. People will buy this technology. You can sell it to visionaries. Visionaries can see where you're trying to go, and you will be able to sell it to them. You've then got the pragmatists, people who are interested and will buy. You do have conservatives. I would describe the NHS as a conservative. That they really want technologies very. When I say the NHS, the National Health Service in the UK is a kind of conservative organisation. They really do want health economics and the whole return on investment proven out. I get it. And then you have skeptics, which are people who are going to be the last to biotechnology, or maybe even never. The point is there's a big gap between the visionaries and the pragmatists, and so you need to start solving this business problem really early on, because you can have early sales, but then you can fall into this chasm. This chasm was defined in a book by a guy called Geoffrey Moore, Crossing the Chasm. And so you've got to have eyes on that chasm really early on. So people have their eyes on technology and manufacturing. they may even get to market, but then they fall down this chasm, and that's the problem that you need to be solving much earlier on. And that's why we have this kind of whole platform of sensors, hardware, apps, which you can, get your smartphones out if you want to download the apps, and also logistics, how you're actually going to get this product to the market. So we have an app on the Google Play Store. So this is a really, I'd call this like a deployment app, because it will get the app onto your, essentially, call them customers, call them collaborators' phones. So this will work for the Android, and this will work for the iOS system. So I'll just go back. So if you've got an Android phone, pause and scan that. If you've got an Apple phone, please pause and scan that. And what you'll find out is when you open the app, it wants to access your phone's camera, because this particular questioner is actually quite interested in multiple analytes. And so what this allows them to do then is the app, in fact, I've got the app. Yeah, every time you scan a code, it will run a different type of assay. So you can run all these different codes. And we do then have a QR code for things that we haven't yet done or haven't yet released. So I would do a little experiment, and I would suggest that you put the app on your phone and then scan these different QR codes, and you can just see that you can go from target to target or analyte to analyte. It's also worth saying, if you've got any more questions around this, definitely sign up for the webinar on the 26th of March. There you'll see a live demonstration from our lab of this hardware platform. The 2 sessions in the day are repeats. So depending on your availability, come to the morning or come to the afternoon session. Question number 2 is about soil health. People are very interested in, obviously, environment. At the same time, farmers are quite interested in saving money. They're indiscriminately putting fertilizers, not indiscriminately, they're trying to be judicial about it, because farmers, one thing they don't want to do is waste money. But obviously there's something wrong with the fertilization process of fertigation of soil, because it all ends up in the rivers. You get something called eutrophication, where all the algae grows, and then the algae drops the oxygen, and you see the dead fish on the surface. So people are interested in soil health, and that, maintaining the health of the soil can help reduce the use of fertilizers. It can also be a source of a capture for carbon, I should say, as well. So it can help with some bigger problems. It's the same situation that actually this one system today can do. We have already launched nitrate and potassium on the system. So take a really careful look at that QR code system I showed earlier on, because if given a sample of nitrate, or a sample and it's got nitrate and potassium in it, we can actually measure it, and we have been shipping that. Again, it delivers all the data to the cloud, so people can start building their kind of cloud offering on top of this hardware platform. So again, you can scan this QR code. It's the same one as earlier on. So if you've got an Android, you can scan the Android. If you've got an iOS, an Apple phone, you can scan this. There's the nitrate app. There's the potassium app. For that, again, it's worth coming on the 26th of March. In fact, we do a potassium demonstration as part of that. The 2 sessions are repeats. We do 2 sessions because some people are in Asia and more towards Eastern Europe, and some people are sort of in Western Europe and into the Americas, Canada, or North America, Central America, and the South America. So we do 2 in the day, because obviously then we can help to cover the time zone quite well. The person here is asking whether we make our glucose sensors commercially available. So the quick answer is, at ZP, we have a lot of CGM, continuous glucose monitoring. So this is almost 2 ends of the spectrum. I've talked a lot about point of need, sometimes called IVD testing. Now I'm transferring over to wearables. The biggest wearable biosensor on the market is the CGM, the continuous glucose monitor. And these are worn on the skin, so I have, the skin has the epidermis, the dermis, and then the subcutaneous tissue. The FDA have just actually done an article where they've said to people, don't rely on non-invasive glucose monitoring. I think they sort of say, like, if it doesn't break the skin, then the FDA is not super interested in it. So, for example, these kind of smartwatches have LEDs on the back of them. Now, Apple doesn't have a commercial non-invasive glucose monitor. They're interested, but they haven't done it, or they haven't released anything. But the FDA is kind of, I realize if you go on, you can look on Amazon and you'll see these non-invasive glucose sensors there. It's really bad because there's no way, this is a strong term, you've got to be careful, but there's a high degree of certainty that they don't actually work. Now, you can do glucose monitoring on a person using transcutaneous or wire electrodes like this. This is what is primarily used. You see people walking around with little circular patches on their arms. These are those kind of sensors. At ZP, actually, we do have a lot of IP, intellectual property, around microneedles for minimally invasive glucose monitoring, but we do break the skin, and therefore we kind of, this is the direction that the FDA thinks is more likely. Don't do glucose in sweat. There is a company doing it, but it's a very hard clinical proof. You have to really prove that out. We do also do implanted sensors. We don't do implanted sensors at the moment for human health. These primarily get put into, it sounds ridiculous, but fish and pigs. But the question was whether any of this technology is actually available, and the quick answer is it is. Just FYI, these are actually a couple of fish that are generally swimming around with some of our CGM sensor technology actually in them. But the quick answer is sure, some of our wire sensors are available on the web store, and here's a link here, and you can go there and have a look, let's say. I'm going to slightly jump now because some of the questions that came in as well were regarding formulations and were sort of more technical. So at ZP, the question came in, what's the cross-linker that we use and whether we use ferricyanide. Just FYI, we do use ferricyanide quite a lot at ZP, but not in the application that you were particularly interested in. At ZP, we make generation 1 and generation, I call them 1.5 sensors. So we do have an enzyme, we do have a polymer. We do encapsulate the enzyme within these polymers, but I can't mention the actual formulation because I would consider that proprietary. It's probably worth saying that anyone who comes to any of our courses ends up making glucose sensors actually on those courses. We do use a barrier layer. I'm not going to discuss why the barrier layer today, but we do actually use a barrier layer. And so we're quite used to building these sort of type, or the generation 1 glucose sensors where oxygen reacts with the enzyme's reacting with the glucose, and we end up forming hydrogen peroxide, and we end up detecting the hydrogen peroxide, and we get a flow of current. We also, I did say, we also do transdermal sensors. I think the part that we've made the most investment is in these actual glucose microneedle watch sensors. There's been at least 20 million euros spent on this. It's quite it's fairly mature technology, at least mechanically and electronically as well, I should say. And that is something that we do have as well. And there's the microneedle patch on the background there. So somebody wants to do multi-analyte detection. The multi-analytes, some of these are already in our portfolio. Some of the ones you're trying to target are extremely doable by the sort of science that we do. I really think that possibly your biggest issue is that you're focusing on the technology. Technology stacks already exist. I think it's thinking more about the business side of things. And in the end, all effort takes money. So you can kind of bootstrap it for a while, but there's a point at which you actually have to raise money so that you can actually push this forward, let's say, with greater momentum. I mean, it's probably worth saying that, at ZP, we'll do efforts with companies, and we'll put something like 10,000 hours in a year or 20,000 hours in a year, so the working hours that you have available to you per year, if you're doing a normal working week, is something like 1,600 hours. So you can see that, a typical program with ZP, could have 8 to 16 people on it, and then so, otherwise, it'll take you 5 years, whereas it could take a group like that, well, in fact, I would actually say that 5 years times that by 16, it would really take you 80 years to sort of do this kind of stuff, and at which point you'd sort of die. So you do need to find funding and bring a team on. Soil health, sure, we actually have the nitrate, phosphate and potassium sensors already. we have already put nitrate and potassium onto the SenseIt platform. Yes, the answer to your question is the CGMs from ZP are commercially available. You do need to be well funded in order to really get involved in continuous glucose monitoring, especially on people. These kind of sensors where they clip into these, we have that kind of technology. This is Dexcom's technology. Dexcom will have a lot of patents around that, so I wouldn't design to their connector because they'll have a lot of IP around that. In terms of formulations, sure, we are immobilizing the enzymes. I can't say what the cross-linker is. We do use ferric cyanide quite a bit at ZP, but we don't use it in the application that you're otherwise interested in. So I want to say thank you very much. I appreciate that. I've been talking almost constantly now for 30 minutes and 30 minutes and 36 seconds. If you've got any questions of ZP, I would say don't hesitate to reach out to us. Okay, thanks very much. And don't forget, there is that webinar, Sense It All, on the 26th of March, 1 in the morning, 1 in the afternoon. They're both repeats, but we just ask you to choose the time zone that works best for you. Okay, thanks very much.